Hey guys, welcome back to Cognitive Science. This week's topic is going to cover the senses. You'll notice that on your syllabus I have divided sensation and perception. Usually people sort of lump them into one lump of a topic, right? I take a little bit of a different approach, and here's the reason why. So, when we talk about the senses, we're focusing on how the brain receives information from the world around it. Okay, now, how the brain processes that information is perception. They're two distinct fields of study, if you will, I suppose. They're two distinct topics, and they deserve their own week's worth of discussion, if you will. So think about in the context of our computer analogy, the keyboard, the computer's camera, the microphone, those are all the tools with which your computer gathers input from its environment, right? And the processor is sort of analogous to perception. So when we think about the computer analogy as applied to sensation and perception, it works really well, right? Okay, so to talk about perception and illusions and all that cool stuff, we need to talk about sensation. And we're going to start by focusing on vision. And we're probably going to spend a little bit more time on vision than, actually, we are going to spend more time on vision than any of the other senses. And that's kind of because vision is the sense to rule all senses, except for maybe like proprioception. But even then, I think that vision wins. And we'll talk about what proprioception is later. So, to demonstrate the power of vision, I wanted to start with this little phenomenon. So, phantom limb phenomenon. This is when people, amputees, okay, veterans, they get painful sensations that seem to originate in a limb that is no longer present. Okay, so they get these weird sensations like burning, aching, throbbing, crushing, shooting pain. And it's weird because this pain, this discomfort is coming from a limb that is no longer there. So, a recent theory by Catalan in 2018, after an amputation, basically thinks that neural circuitry related to the missing limb loses its role and becomes susceptible to entanglement with other neural networks. So, in this case, the network responsible for pain perception. So, I don't know if that makes sense. So the nerves that are connected to this amputated limb no longer have a connection, and they accidentally get tangled up with other nerves, and this entanglement causes these odd sensations. So imagine that you lose your hand. Okay, it leaves sort of a big chunk of real estate in your brain and in your nervous system as a whole that it just doesn't have a job. It doesn't have anything to do because it no longer has a limb to control. So it stops processing any kind of sensory input, it stops producing any motor output to move the hand. It goes kind of like idle. But it doesn't necessarily go silent. Okay, neurons are never completely silent. So when they're not processing a particular job, they might fire at random. Okay, and this sort of coincidental random firing of neurons in that part of the sensory motor network can sort of happen at the same time as from the network of pain perception. So when these networks fire together, they create sort of this experience of pain in that part of the body. So it's this coincidental firing of the cortex that handles your hand that is combined with the firing of a pain network. And so you think that you have pain in a hand that is not actually there. So, how do we help people with this particular affliction? Okay, what does this have to do with vision? So, we use what's called a mirror box treatment for people with phantom limb, and it helps quite a lot, actually. So, essentially, you have this box, and okay, so here is the amputated limb, right? Well, this isn't an amputated limb. Maybe it's amputated past past this point here. Anyway, okay, so his amputated limb is in this side, his intact limb is in this side, and there is a mirror here. Okay? And what he's doing is he's looking on this side of the partition. Okay? So what he can see is that he's got a hand here and he has a hand here from where he's looking at the mirror. Does that make sense? So you can kind of sort of tell what I'm talking about in this particular image. So if you imagine that the hand is gone, but then you have this hand in front of the mirror, so then you're looking at the mirror, and if this hand were connected right here, it would look like you've got your other hand, right? Okay, so the therapy works because the visual system is kind of the boss. And when these patients look at this partition and they see their amputated hand sort of completed again, okay, the neural confusion that they have can kind of get explained by their visual system. Okay, and eventually the brain starts to understand what's really going on. So we correct this very non-visual perceptual anomaly with vision. Okay, because vision, the whole like, oh, I can't believe my eyes, the saying should actually be, you know, I can't not believe my eyes. You have a really hard time not believing what your eyes are telling you. Okay, and that's why illusions can be so fun. Okay. So, when we talk about vision, we're going to talk about vision at the level of the eye, and we're going to talk about vision at the level of the brain. So we're going to start now with talking about vision at the level of the eye. So here's your eye. Okay, basic eye structure. We'll go over a couple of basic structures so you understand how light is getting into the eye and how it's being transduced into neural signal. And we'll talk about what transduced means in a little bit. Okay, so we'll start with the cornea. The cornea is this hill here. Okay, it's the bumpy part of your eye, and what it does is it refracts light down onto the lens in a particular way that you need, right? So you can see the way that the cornea is curved. It's going to shoot light into the pupil, onto the lens where it needs to be. Okay. The pupil is not actually a thing. It's a hole in a thing. The pupil is like the donut hole. The iris is like the donut. Okay, so here's the iris. It's like a couple layers of pigmented cells, and that's what gives you your eye color. And the iris sort of constricts back and forth, and this is what causes change in pupil dilation. So here is an example of a dilated and non-dilated pupil. The diameter typically ranges anywhere from 2 to 8 millimeters. That is a 16-fold increase in the amount of light entering the eye from 2 to 8 millimeters. The pupil itself doesn't dilate. The iris dilates. So when we talk about, oh, your pupils are dilated, it's more like your iris is dilated, making the hole in the middle of the iris larger. Okay. Why do we have pigmented cells in the iris? It's to prevent light from getting in places other than at the location of the pupil. So let's go back to this here. So we've got light refraction going on here, bending light down into the pupil. The pupil directs light onto the lens, and the lens is going to take it straight back to the retina. Okay, so if we have light coming in at all these different places here as well, okay, this is going to cause issues in here. We're going to have a little bit of a problem focusing the light where it needs to be focused in the eye. And you'll see why that can cause issues later. All right. So the lens. The lens is this guy here. It's just like a lens on your glasses, a lens on a camera, except this lens changes shape to change focal distance. So when you're looking at something far away, your lens changes shape to accommodate for the distance that light is coming from, versus when you're looking at something closer, right? It has to refract it differently because it has to project it back onto this point in your retina consistently, right? It has to hit the back of your retina consistently. So let's look at what I'm talking about here. Okay, so when we have what we call ametropia, that's normal. Okay, this is normal vision. So we have light coming in, it hits the lens, it's refracted nicely and hits the retina right where it's supposed to hit the retina, right? Everything comes together right on the retina. Cool. All right. Now, myopia is nearsightedness. So you see things better when they're up close. Okay, you are sighted for nearness, which means far things are blurry. Okay, so when things are far away, they come in, they hit the lens here. Okay, and the lens is not the shape that it needs to be, right? So what happens is it hits here. Okay, now when it hits here, you're not going to have complete focus by the time it actually hits the retina. Do you see how the light is scattered out by the time it hits the retina? Okay. Now, myopia with correction, so you've got your glasses on, you've got your contacts on, they're assisting the lens so that refraction can hit right back here on the retina. Hyperopia, okay, this is farsighted. You see far away things better than things up close. Why? Because your lens over refracts, if you will, so it sends the image further back than your retina is, right? So by the time it hits your retina, it's not quite converged into its one image yet. Okay, so you could do the same thing, hyperopia with correction. Okay, and this will assist the lens in refracting the way that it needs to refract. Okay. What leads to these particular issues? It's kind of, it has to do with the length of the eye. Okay, so look at the length of the eye here in ametropia. We've got a nice little circle, nice little round eyeball. And here in myopia, right, the eye is elongated. You can tell that the eye is not quite a circle. And here in hyperopia, the eye is sort of squished. It's a little squishy circle. Okay? All right. Now, let's talk about where the magic happens. This is at the retina. Okay, this is where things get fun. So the retina has all the photoreceptors, and the photoreceptors deal with light transduction. Transduction is a big word in sensation and perception, okay, and it's one that you should know. Wink, wink, nudge, nudge. Okay, so transduction, your neurons don't sense light. They don't sense sound. They don't sense any of that. Okay, they sense neural signals. So we have to translate what we have in the environment around us into neural signals so that our brain can sort of make sense of it all. Okay, that's transduction. At some level in every sensory system, there is a group of receptors that is responsible for transduction. These receptors take input from the environment and transform it magically, not magically, we'll just say that for now, into neural signal. Okay. Right, so this is, you remember Cajal, we talked about his staining methods. He took the Golgi stain and he perfected it all, and we figured out that neurons don't actually touch each other. And yeah, okay, so he made some really cool drawings, and this is his representation of the retinal layers. Okay, so, okay. First thing to know is that these are the rods and cones, okay, up here, rods and cones. You can tell, see, kind of cone shape, kind of rod shaped. All right, those are photoreceptors. Light is coming in from here. Photoreceptors are at the back of the retina. Okay, so this is the part that's facing outward toward the eye. Okay, so the light is being sent all the way back to these photoreceptors back here. Okay? All right. So let's look a little bit closer at what's going on here. All right, so in the retina, we have, what are we starting with? Right, rods and cones. So we got rods on the left, cones on the right, as you can see by their shape. Rods have a lower threshold for light. Okay, remember we were talking about firing thresholds. Your neuron is resting, it's resting, it's resting, it's calculating, it's calculating. Oh, it hits a threshold and it fires. All right, well, the threshold for rods is lower for light. It doesn't take as much light to activate a rod. Okay? That means that rods are good in low light scenarios. Okay? Because they are more sensitive to light, this also makes them better for motion perception. Okay, so those are that's what the rods are specialized for. Okay, they're specialized for low light scenarios, motion perception. Okay, and they are very prevalent in peripheral vision, which is why peripheral vision isn't as you don't have as much acuity in peripheral vision as you do in central vision. Okay, so the cones, these guys over here shaped like cones, they're less sensitive. They need more light. Okay, they have really high spatial acuity. Okay, so if you want to look at something in detail, what do? You turn on the light, right? You really want to get at all the little nooks and crannies of whatever it is that you're looking at. You're going to be using your cones. The super high spatial acuity, they're really good at spotting detail. They mediate color vision. Okay, so rods don't really have a lot to do with color vision. They're more of a black and white kind of people. Cones are all about, they like color, different colors, right? And this is what's going to help you see detail. The more color you can see, the more detail you can see. Cones are highly focused at the fovea of the eye. So we're gonna talk about that in just a second. I think I've gotten ahead of myself here. So let's review. Rods are more sensitive, better in low light, better with motion. Cones mediate color vision, high acuity. All right. The fovea. I did get ahead of myself. Looky here. Here it is. This is the center of your visual field. Okay, now this is the optic nerve. Why isn't the optic nerve the center? We'll talk about that in a minute. The fovea is the center of your visual field. When you're really examining something in great detail, you're relying on that fovea. It's got the highest concentration of cones out of all of your retina. The very center of the fovea has only cones. Okay. So now, what happens from here? Oh, so you can see here, these are rods, these are cones. This is non-foveal, right? This is more out in the periphery, maybe not completely in the periphery. And this is a foveal representation of what's going on. So this is the fovea, cones in the fovea. So this is all cones. This is the eccentricity from the fovea. So if you imagine the fovea is here, and this is further out into the periphery on the retina, so you're going further away from the fovea on the retina, this is the amount of cones, right? So lots of cones in the fovea, not so many cones at all out in the periphery. Okay. All right, so the light goes through all of this stuff. Wait a minute. Okay, so light goes through all this stuff here, okay? And what it does is it has to come back out to the optic nerve, right? So the rods and cones, they get all this information, they send it to these ganglion cells, okay? And the ganglion cell nerve fibers actually are optic nerve fibers, okay? The optic nerve is composed of ganglion axons. All right. Now, I think I have something for that later, but forgive me. Now, when the light comes back out, okay, that information is being sent along those ganglion axons and the optic nerve. We talked about the optic nerve, how we have left visual field and right visual field information coming in. Okay, so the optic nerve gets all this information. The problem is that there's no room for photoreceptors here. This is all nerve. All the axons converge in this one spot. So what does that mean? If there are no photoreceptors of that part of your retina, it means that you can't see anything if light were to be directed at that spot on your retina. It means you have a blind spot. Yeah, you probably don't believe me because you don't have a blind spot when you look around at the world. You don't have like this little, black spot around it. Oh, that's my little blind spot. That's my friend. We don't have the blind spot when we're looking around regularly. Okay, but try this. Okay, you're gonna close your right eye. Okay, look at the fixation cross with your left eye. Keep looking at the cross and move your head closer and farther away from the screen until that dot disappears. You will find the spot where that dot disappears. Okay, you're cover it up, and you need to be using your left eye if you're looking at the fixation, because we're putting that dot in the periphery on the left. Okay, so right. Okay, so cover up your right eye, looking at the cross. Move to the point where you're looking at the cross where that spot disappears. You won't see it on the slide. I'll give you a minute to check this out, or you can pause it right here. Okay. Luckily, vision is really great at filling in the blanks. And this comes at the level of perception, not at the level of sensation. Okay? This is why these two processes are it's so important to separate them. All right. We also know that vision is really good at filling in the blanks because of things like floaters. Okay, we all have there's like, okay, so the stuff in your eye is called the vitreous humor. Okay, it's just liquid in your eye. And there's stuff floating around in that liquid. There's just particles of debris, little tiny things floating around in there. It's not a big deal. Everybody has it. Don't worry about it. We don't see them. You don't ever see, well, sometimes you do. When you see floaters in vision, it's because it gets a little complicated, but because of overfiring of photoreceptors, and they become fatigued, and so you can start to see different things that you shouldn't necessarily be seeing. Okay, so when you see stars, right, you're seeing neural firing, actually, when you see stars in your visual field. Okay. The biggest thing that I want you to remember is that photoreceptors don't see color. Okay, photoreceptors don't see color. I know that I told you that cones like color and rods and cones like black and white, okay, but cones like the particular wavelengths of light that are associated with what we know to be color. Okay? Color does not exist in our environment. Waves of light exist in our environment. Our machinery interprets those waves of light to be color. All right. We talked about things at the level of the eye. Now let's talk about vision at the level of the brain. Not perceptually, but just how do we get that signal once it's transduced? How does it get on back to the brain? Okay? And that's going to be in the next lecture, okay? But it's basically going to be when we talked about left visual fields, right visual fields, and how they cross at the optic chiasm, right? So we're just going to review that stuff in vision and the brain. So I will see you there.